Hello. I guess it is time to start. You're welcome to the World Potato Congress webinar. And that first slide is the announcement on the 11th WPC Congress 2021 in Ireland. I'm Leah Tsror from the Agricultural Research Organization, which is under the Ministry of Agriculture in Israel. And probably, as you have noticed, I'm delivering this webinar from home because of the corona pandemic. I'm at home. We are not allowed to go to work. We're allowed only to go to the supermarkets, and that's it. But I guess we are not the only one in Israel. A lot of people all over the world are at the same situation, and hopefully we will get over it. So now we will start with the webinar of today, and I'm pleased to present it. The topic is Powdery Scab, Integrated Disease Management for Reducing the Risk. In my talk, I will give a short background on the disease and the pathogen. I will discuss several epidemiological aspects as environmental conditions, alternative host survival and spread, and latent seed infection, and then get into management practices. I will try to cover several approaches, preventive measures, cultural sustainable control, and chemical control. So the powdery scab actually is a worldwide disease affecting seed and wear crops, causing severe damage all over. The estimated annual losses in UK is about 7 to 9 million pounds in wet years, according to the British Potato Council. The symptoms of powdery scab are lesions, scab lesions on the tubers, as you can see here, and the scab lesion contain powder of spores. From that is the name of the disease. Now, these lesions downgrade the quality of tubers for fresh market, processing, or storage, and specifically, it may cause rejection of contaminated seed lots. At the bottom, you can see the cankerous form, which is the most damaging symptom because these kind of tubers are rejected for any use. In addition to the tuber lesions, the symptoms include also galls on the stolons and also on the roots. Now, the galls on the roots may interrupt nutrient and water uptake, leading to yield reduction. It was reported from New Zealand and Australia. However, in Israel, we do not face any yield reduction due to root galling. And also from Washington State, Dennis Johnson reported that there is no yield reduction. However, if other soil-borne pathogens are present, such as, for example, Rhizoctonia solani or Coleotrochum cocoides or nematodes, and root rotting occur, then it might also lead to yield reduction. The causal organism is Spongospore subterranea, which is a plasmodiophorid from the kingdom Protozoa. It is an obligate parasite which can only be cultivated on host plants, which makes it very difficult to work with and indeed a great challenge to all research groups who are working on this disease. And as I mentioned earlier, in the scab on tubers or in the galls on the stolon and roots, there are sporozoa, or they were called before spore balls, and each of them contains more than 100 resting spores. And I will shortly explain the disease cycle. These resting spores, which are actually the survival stage, under the suitable conditions, which include a host and high soil moisture and low soil temperature. Then a primary zoospore will develop and will swim in the free water that must be in the soil and will infect either directly the roots or the tubers. And this is the infecting stage in the root, producing plasmodium, zoosporangium, which will release secondary zoospores, which can reinfect the roots or the tubers. It means it is a polycyclic disease, and with a very low inoculum under the suitable conditions may create a severe disease. When the conditions are less suitable, new resting spores will be formed on the tubers and also in the galls on the roots. The tuber infection mainly occurs during the tuber initiation, while the root infection continues throughout the whole season. Spongospore is also the vector of mop-top virus, which suppress the plant growth and cause internal tuber necrosis. In Israel, we don't have mop-top virus. Now you see on the left tubers with powdery scab, and on the right, common scab. It is very easy to confuse between these two scab symptoms. And for example, this tuber, is it powdery scab or common scab? It means that it is not possible to diagnose only according to the symptoms. Therefore, we must use methods for disease diagnosis, which include microscopy, as I've already mentioned, serological tests. There are kits that are available and are quite sensitive and easy to use. But in the research, we use molecular testing, either as in PCR analysis or RT-PCR, after, of course, DNA extraction, and we use specific primers. It is a very sensitive method. Now I will refer to several epidemiological aspects. First, the inoculum. The pathogen is tuber-borne and soil-borne. And of course, it implicates on the management. The environmental conditions I've already mentioned. The zoospore will develop only in the presence of a host. There is a narrow window for the tuber infection with low temperature of 12 Celsius degrees, while the infection of the roots may occur in higher temperature and along the whole season. Sporozoa survive in soil and of course also on plant residues. They survive also in animal manure. The spores are resistant to the digestive system. And of course, volunteer potato plants are a very good bridge. The sporozoa can spread by runoff water, machinery or implements that have soil particles, and also by wind. Alternative hosts, there are weeds and rotational crops that may serve as alternative hosts, and I will elaborate on that. Now in Israel, we have faced disease outbreak in potatoes that were grown for the first time, first crop in a new uncultivated field, actually virgin soil, and that led us to the hypothesis that Spongospora is spread by wind. Therefore, we have positioned wind and soil traps in different locations, as you can see here, for example, in this pivot center plot and this one, the red dots are where we positioned our traps. And indeed, we managed to capture Spongospora in the ground traps as well as in the wind traps. It means that indeed it can be spread by wind. Moreover, we sampled soil in an uncultivated area just near the potato field, and we were able to detect the pathogen in 300 and even more, up to 700 meters. So certainly, the spread of Spongospora by wind plays an important role in the disease epidemiology. The host range of Spongospora was investigated by several research groups, and we had research on that as well. We focused on finding natural infections. So we sampled plants in commercial potato fields with powdery scab history, and we sampled weeds and rotational crops as well as potato volunteer plants. We also made some experiments with artificial inoculation of weeds and rotational crops. And as you can see here, they collected the plants or the inoculated ones. The root system were thoroughly washed. And then from the same root system, part of it was taken for a microscopical observation after staining, or DNA was extracted and real-time PCR analysis was performed. And indeed, in the natural infections, we were able to find 16 species out of 52 that we collected, among them Solanum species that were already reported as naturally infested, but also some other species. Among the rotational crops, we were able to find positive wheat plants, but barley plants were negative. And of course, potato volunteer plants were positive. In the artificial inoculation of the weeds, we were able to detect 13 out of 16 species positive. Among them, of course, the Solanum nigrum, which had the highest severity of infection. And also with artificial inoculation of rotational crops, we managed to get 11 out of 14 rotational crops positive to Spongospora. Of course, potato and tomato, which is also a host for Spongospora, although it's not rotated in Israel with potato. Radish, you see very high infection. And it's interesting because we managed to inoculate barley, but we couldn't find it in the naturally infected one. Maybe the size of the sample was too small. This wide host range may increase or at least maintain the inoculum in the soil. Now for the disease management that can be applied. So first, the preventing measures, which concentrate on pathogen-free seed tubers and soil and also manure, and the use of resistant cultivars. I will elaborate on that in a few minutes. And of course, reducing the inoculum sources, either by cultural treatments, the planting date, foliage cover, and fertilizers. I will refer to that in several slides. Biological control agents. Aspergillus was reported from Japan as an effective treatment which reduced powdery scab at 54 to 70%. There are also reports on trichoderma and bacillus as biocontrol agents. However, these treatments are not consistent, and as far as I know, there is no commercial material that is targeted to control powdery scab. Biofumigation, soil treatments, and seed tuber treatments are also measures to reduce the inoculum, and I will refer to it very shortly. Among the agricultural factors, long crop rotation, although it is not highly effective because the pathogen may survive during 13 or even more in the soil. Avoiding soil compaction and waterlogging, sanitation and hygiene during grading or storage, and of course, optimal irrigation and fertilization. So now we will continue with the preventing measures, and that is the first, monitoring disease in the seed tubers. In Israel, we monitor blemish diseases in the seedlots that we import every spring from Northern Europe in sample of 200 tubers per lot. And you see here the figures for black dot, for silver scurf, and for common scab. You see that among the imported seed material, which all are certified seed tubers according to the Israel permit, there are quite a lot of infected. I'm back with you. So I will go very quickly on the monitoring the blemish diseases in the seedlots. And the last sentence was that unfortunately, so far, there is no effective seed treatment available. In the past, contaminated seed tubers were treated with mercury or formaldehyde, but of course, these nasty materials are forbidden. Now, knowing which fields are contaminated with powdery scab can assist in making a right decision on site selection, and soil testing is important in order not to count on the memory of the grower about his powdery scab field history. So a protocol for soil sampling, DNA extraction, and real-time PCR was developed in Scotland, and we applied it in a survey that we have done. You can see here from 2012 up to 2015. The red part of the columns are plots that were contaminated with more than 10 sporozoa per gram soil. The gray were up to 10, and the orange one were the clean ones. And here you can see the correlation between soil inoculum and disease incidence in the field. It means that the correlation is not very high. Nevertheless, soil testing provides a very important information on the potential risk. But the disease development actually depends on the environmental conditions. And the other preventive measure, which is maybe the most important, is using a resistant cultivar. And also in Israel, as in many other countries, we have evaluated a lot of commercial varieties under naturally infested soil condition in field trials. And as you can see here, just an example from one of the trials. Valor, for example, is a tolerant variety with almost no powdery scab symptoms. And on the other hand, Nicola, which is susceptible in Israel, or Charlotte, with very clear, severe symptoms of powdery scab. In the next slide, just 1 minute, we use in our evaluation the international scale for assessment of powdery scab tuberous lesions, as you see here. And we also evaluate the root galling in our trials. In this slide, we can see the average powdery scab incidence on the tubers on selected cultivars that were tested at least in 2 trials and more. There are some cultivars here that were tested even in 6 trials. And as you can see here, Nicola is the susceptible variety with about 50% disease incidence, and Valor, the tolerant one, with about 15% disease incidence. In the next slide, you can see the disease incidence of the susceptible, the red ones, Nicola or Allianz, and the tolerant ones, Valor or Winston, during the years that they were included in the test. And we can see that there are variations across years. In 2008, 13, and 16, where the disease incidence is quite high, the winters were colder than in 2011, 14, or 17. And all trials, I just remind you, were conducted in highly infested fields. Now, in addition to the assessment of the cultivars to the tuber scab, it's very important to evaluate the response also for root galling, because root galling may affect the buildup of the soil inoculum and sometimes to reduce yield. And as you can see here, in the tolerant variety, both the tuber lesions and also root galling are low. In the susceptible variety, both are high. And in the other varieties that are presented here, we see some variations. For example, cultivar Dita, which has low, not low, but medium scab on tubers, but high root galling, and the opposite in Marispir with high scab lesions on the tuber and lower root galling. So it is variable, and it is important to have the data on both scab lesions and root galling. Now, from the previous slide that I mentioned, the variation across years, probably because of temperature, because in Israel, even the winter crops are irrigated, because in the area where 95% of the potato production in Israel, the annual rainfall is about 200 millimeters, and that is in good years. So irrigation means that soil moisture is available. So the temperature may be the crucial factor. So we ran an experiment with different planting dates, 7 of October, 25, and mid-November. And these are the results of this experiment. The red columns are the disease incidence of the 15th of November planting date. And the black dots are the disease severity. You can see very clearly that the mid-November planting date had significantly higher disease incidence and also severity. So it means that planting at a warmer temperature may reduce the disease impact. And indeed, if we look on the temperature, you see that the minimum temperature and also maximum temperature during tuber initiation time, the mid-November planting was much colder than the October planting. So it implies the temperature indeed plays an important role in the disease development. Now, taking this factor, we have tried to use application of non-woven fabric in order to increase temperature. And I will show you some slides. This is the fabric. It is a breathable fabric, and we cover the rows 1 week or so after planting, and it remains there for about 60 to 70 days. You see the foliage bring up the agaril net as the crop develops. And indeed, you see here the maximum and minimal temperature. We set the data loggers in depths of 15 centimeters in the soil, and the temperature in the agaril net was by 2.5 to 4 degrees higher than the uncovered area. So it means indeed that we can manage to increase temperature by this treatment. And by the way, this approach is suitable also for the organic sector, of course. Now I will show some other treatments that we have tested in our trials. Pre-planting application of a pelka, which is actually a fertilizer. In the process of decomposition of the pelka in the soil, cyanamide is released, and this is the toxic fraction which affect microorganisms in the soil. And of course, I will refer also to chemical treatments. All of them are pre-planting, either methampsodium or chloropicrin soil fumigation. And please remember the plastic cover in the chloropicrin application, broad or in furrow application of fungicides. And all this now I will present you some results of our trials. The recent trials where we tested the foliage cover and some soil treatments. You can see here the disease incidence in the control, and the disease incidence was reduced by 90% in the agaril treatment. It is the cover, the plant cover, also by methampsodium that was applied by overhead irrigation, by the fertilizer pelka, and also by fluazinum, which was applied by broadcast application. The methampsodium shunt injection had the probably technical problem, and that is the reason why it was not so successful. These treatments had a significant effect also on the root galling. As you can see here, this is the disease incidence, the root galling incidence in the control, very high incidence, and it was significantly reduced by all treatments. And pay attention to the plant cover with a non-woven fabric. It is almost similar to the methampsodium application and the fertilizer. Now, in this specific year, the agaril net resulted with higher yield because of a frost event that occurred 60 days after planting. So all the plants in all treatments were damaged very badly, except for these plants that were covered. But as I mentioned earlier, usually we do not face any yield reduction at all by powdery scab. And that is another field trial where we tested again the agaril plant cover that reduced disease incidence by 70%, fluazinum applied by broadcast, 10 liters per hectares, the 5 liter per hectare was a little bit less effective. However, significant reductions were obtained by all treatments except the nebigine flusulfamide, which is a registered agent that is used in New Zealand and Australia. It was not effective here. Now, the root galling was very low, except the control and the nebigine. Another treatment, soil treatment that I wanted to show you is the soil fumigation. And that is the metam sodium with overhead, applied by overhead irrigation, which I've shown you already that is very effective. This is the chloropicrin that was applied with the plastic cover. When the chloropicrin was applied without the plastic cover, there was no effect on powdery scab. So we had 90 to 98% reduction by the chloropicrin with the plastic and also by the metam sodium application. Root galling was about 40% in the control and 0 in the chloropicrin covered treatment and metam sodium application. And that is an important point which I want to show you. This is the inoculum levels in the soil. The blue columns are just after the fumigation, and you see this is the level in the control, which was decreased significantly by all chloropicrin and metam sodium treatments. But pay attention, after harvest, the levels in all treatments increased. It means that within 3 months, the inoculum is increased, and actually the soil treatments have only a short-term effect on powdery scab control. Biofumigation is another alternative, sustainable, organically. It is the use of brassica crops as green manure that is known to reduce soil-borne pathogens due to the production of volatile sulfur compounds, which are toxic to soil microorganisms. And indeed, in a paper that was published a long time ago, in an on-farm biofumigation field trial, Indian mustard, rapeseed, and canola reduced powdery scab in the potato grown afterwards by 15 to 40%. So we have tried this also in Israel in a field trial on an infested soil. We tested 2 cultivars of mustard, Red Giant and Terminator, but you see here that it was a failure. We didn't get any effect on powdery scab, not by the brassica treatments, but also not by the metam sodium treatment. And this is probably due to accelerated biodegradation of methyl isothiocyanate, which is the major toxic volatile, which is released in the biofumigation process and also in the metam sodium fumigation. And that is a phenomenon that we face in Israel, mostly in plots that are treated several times with metam sodium. So probably further research has to be done, but this is quite a promising approach. Now I would like to summarize, and indeed we have to take into account several measures in order to have a rational powdery scab management. And starting with the cultivar and field selection. The cultivar choice is important, and in assessment of cultivars, powdery scab on tubers and root galling as well should be evaluated. Use of pathogen-free seed tubers, of course, is a must. because no seed treatments are available so far, and pathogen-free soil. By soil testing will assist to select the right field. And of course, to prevent weeds and volunteer potato plants. Among the cultural sustainable measures that can be taken, the planting date, using the plant cover by a grill, Perlka needs further investigations, and in the literature, zinc is also reported as reducing powdery scab, and as I already mentioned, biofumigation and biological control. Among the chemical measures, soil fumigation with methyl sodium or chloropicrin, where it is registered. It is highly effective and especially under high disease pressure. In furrow or broadcast application of fungicides, fluazinum, mainly fluazinum, will be effective under low and medium disease pressure. Among the agricultural practices, we should take into account disinfection of machinery, hygiene of store and grading lines, avoiding plant stress, means optimal irrigation and fertilization, and minimum soil compaction. Under high disease pressure, which means high soil inoculum, susceptible cultivar, and conducive conditions of cool and wet conditions lead to high risk. And here you have a scheme that enables the growers to take a rational decision for powdery scab management. And just thanks for the funding source, which enable us to take all the research in this topic. And thanks the sponsors of the World Potato Congress. And also I'd like to thank you, the participants, for attending this webinar. And now I believe we will have time for questions. I'm ready to take questions. Just a moment, I see that there is in the chat. I'm trying to get into it. Okay. Question one is, what is the level in the soil that is okay to plant into and not cause disease? So actually, as I mentioned before, this is a polycyclic disease. So the inoculum in the soil has to be very low because it was demonstrated that even 5 sporozoa per gram soil is enough to cause symptoms in conditions that the environmental conditions of humidity, soil moisture, and temperature are suitable, and of course the cultivar. It means that the inoculum level should be very low, below 5 sporozoa per gram soil. Another question that I see is powdery scab moving to new production areas around the globe. So I can say from personal impression that I'm sure that the powdery scab in Israel started from the imported seed tubers that imported from Northern Europe, because it is not an endemic disease in Israel, and it is usually was not considered as a pathogen that can even develop in hot climate areas. So the answer is yes, certainly, because of contaminated propagative material. Another question that I see, what is the fungicide that appear to have some activity on powdery scab? So the answer for that is fluazinum. And I think I demonstrated in my slides that fluazinum in high dose, when the disease pressure is high, was quite effective and reduced by 70% the disease on tubers. The next question that I see, can you explain more about the soil test and how is it interpreted? So I guess that the question is more on the how to interpret the results, because technically the soil samples should be taken randomly, that they indeed will represent the field, and a lot of sampling points are required, and then the analysis is done by real-time PCR, which is highly sensitive. The interpretation is quite tricky, because the real-time PCR analysis is very to the point. If the answer is positive, it means that the potential for causing disease is there. It means that if the real-time PCR is positive, the grower has to take into account that his field is contaminated, and therefore he has to take some other measures, either trying to use tolerant variety, or try to manipulate the temperature by planting date, or as I have demonstrated to you, the plant cover in order to increase temperature, etc. Or, of course, to take some more harsh measures and apply some chemical treatments like soil fumigation or fluazinum or other fungicides. The next question is, can you suggest a reference that lists powdery scab resistant susceptible clones? Well, that is difficult, because I don't think that there is one reference for that, but if you will email me, I can send you some references that contains a very informative material regarding these questions. The next question is, what do you think is the best option to assess the disease? If I understand correctly the question, what we are doing is sampling the roots before the foliage is distracted, and to sample the tubers, the daughter tubers, even before horn destruction, and to evaluate the disease using the scales that I've shown. However, as I mentioned earlier, there are also latent infections on the tubers. So if it is very important, seed lot, like mini tubers or so, I would suggest to consider also try to assess the seed lot by peeling and DNA extraction and RT-PCR analysis. I hope that I've answered this question. The next question is, does the PCR method valid for any kind of soil? Oh yes, that is valid, although for each soil type we have our calibration, because each soil type will act differently. So we prepare calibration by inoculating different soil types with known sporozoa numbers, and then we run the real-time PCR, and then when we have a sample from commercial plot, we can compare it to the calibration scale. Well, the next question is, what about spunta susceptibility to powdery scab? Well, I'm not sure, because we have never evaluated spunta. Spunta is not grown in Israel for a long, long time. So sorry, I can't answer this question. The next question, have you looked into possible trap crops? Well, that is a very interesting question, because in our research on alternative hosts, there were some hosts that we were not able to detect sporozoa. Only the sporangium stage in the roots. However, I'm not sure that we just couldn't observe the sporozoa, because if we were completely sure that sporozoa are not formed, it means indeed that certain crops can be trap crops. That means that the Spongospore can germinate, develop, but the stage of forming the sporozoa at the end of the growing of the particular crop is not possible. However, we must be very, very careful with determination about a specific crop, if it is a trap crop or not. The next question. In North America, farmers begin planting seed at 10 degrees centigrade. Your data suggests that waiting to plant until the soil temperature is warmer would help reduce infection. Yes, indeed, it is. And there is a paper by Barbara Christ, long time ago. It was demonstrated in her paper, in a work, that planting later was effective and the powdery scab incidence was lower. It was a paper that was published in American Journal for Potato Research. If you will be interested to get it, email me and I can send it to you by mail or the link. The next question is potential cover crop for powdery scab. Well, I mentioned the cover crops from the brassica, which are supposed to be very effective because their effect by releasing the toxic volatiles, including releasing methyl isothiocyanate, which is effective against powdery scab because methyl sodium is effective. So using brassica cover crops, which have to be incorporated into the soil in order to be effective, I think that this can be a good approach that can work very good, unless the phenomena of biodegradation, as we faced in our trial, is occurs. The next question, how can soil be treated for sustainable production, even if the pathogen is present? Do you think the pre-treatment with biological agent before planting will have control, such as trichoderma or other known biocontrol agents? So actually with biocontrol agents, not only against Spongospore, but also other soil-borne pathogens, the problem with commercial application is that the results are not consistent. And if I refer particularly to powdery scab, the only paper that was published in Japan with aspergillus as effective and reducing powdery scab by 70%, it was applied as seed tuber treatment and not to the soil. However, trichoderma or bacillus that were tested against powdery scab were not consistent. Sometimes they were very effective, but sometimes they were less effective. And this is a consistent problem with also other pathogens such as Rhizoctonia solani. So I think that further research is required also in this aspect. The next question, do you have experience with using compost as a management tool? What role do microbial activity and diversity play in general in terms of interference with powdery scab? So we don't have any experience with compost application as a management tool against Spongospore. However, we do have experience with compost treatments against Rhizoctonia solani, which worked very well, but again, like biocontrol agents, it is depending on the compost quality. If the compost is not mature enough, if the compost is not in good quality, it might be not effective. And on the contrary, if someone will use compost that is not mature, it means that it may even contains pathogens that were not eradicated by the compostation procedure. So a lot of care should be taken in trying to implement compost as a management tool. Although microbial activity and diversity play an important role in general when we refer to soil-borne pathogens, not only Spongospore. So again, it depends. And by the way, the biofumigation is also a procedure that tries to improve microbial activity and diversity, and not only releasing the toxic components. So I think that the answer for that is, yes, it is important, but again, not enough data or findings are available. The next question, except from fluazinum at high dose and chloropicrin, which are not allowed anymore in Western Europe, no other chemical options are available. Well, I think that I don't have very good things to say about that, because fluazinum was demonstrated as effective. And we have tested also flusulfamide, which was reported from New Zealand and Australia as effective, but it was not effective under the conditions in Israel. We have tried in the past also azoxystrobin, which showed some efficacy, and also another soil treatment. I can't remember the agent now. If you email me, I can give you some more details, but fluazinam by far was the most effective one. The next question, do you see any other advantage from the cover crop on crop health? Or perhaps there are some negative effects? Well, the cover crop, I think I've already answered this, because the cover crops contribute a lot to the soil health by enrichment of microbial communities, concentrations, and also diversity. Next question. Globally, there is an increase in powdery scab. What is the situation with moptop virus? It seems that moptop virus is not following powdery scab increase. Well, I'm not a specialist for the moptop virus. We don't have it in Israel. And it might be that the growing condition in Israel do not allow the virus to develop, or I'm not sure about other regions, but as far as I know, in North America, moptop virus is a problem, and in screening cultivars to powdery scab, moptop virus is also a very important issue to look at. The next question is, what dose of Perlka was used in the trials? Well, we used 500 kilogram per hectare, which was effective. An important point, it is a pre-planting treatment, and it should be done at least 2 weeks before planting, and even more, because another week is required for giving time for release of all residues in the soil. Otherwise, it may be phytotoxic. So be very careful with it. The next question. I know that russet Burbank variety does not show powdery scab on the tuber skin, only on root. Does it mean that russet Burbank seeds are already powdery scab free? But, well, I don't know to tell you if it is powdery scab free, but it is recognized, known, reported that the russet types are more tolerant to powdery scab. I'm not sure they are free, but they are more tolerant. Next question, do you have any suggestion for avoiding potato moptop virus be transmitted through powdery scab? Well, the answer for it is that you have to control the vector. Spongospore should be controlled. Otherwise, moptop virus can be developing and expressing symptoms. So I think the answer is you have to fight the vector and not the virus. Next question. Okay, I see it is from Nora, and we will need to end the webinar due to time. Thank you very much, Nora. thank you all the participants. It was indeed a pleasure. And if there are additional questions, please feel free to contact me by email. Thank you. Bye-bye.